We're here at the IAS meeting in Vancouver, BC. We're here with Chris Peterson from the Fred Hutch Cancer Research Center in Seattle, not far from where we live. And we appreciate you taking the time to visit with us. You just finished a press conference addressing the issues towards a cure. And there's so much of that going on today, and it's, as Sharon put it, we're trying to organize it a little bit better, and eventually it'll get to be more, standardized, if you will. But meantime, we want to hear from you and the work you're doing in genetic editing.
Yeah.
Yeah.
So we have a preclinical model that we set up to basically what we call gene editing for stem cells. So we can use a nuclease that's called a zinc finger nuclease, and it's one of a number of nuclease platforms that can basically be introduced into cells and clip out a gene of interest or make a double strand break in a gene of interest. And then when that is induced, then that gene becomes mutated and won't be expressed anymore.
So this is the process by which you can say you're at the early stages, but really hopeful.
Yes.
That it does everything you expect it to do up to this point.
Right.
So.
Yeah, as you can imagine, what I just said is more complicated than maybe I made it sound. But so the first step is just to show that we can do it.
Right.
And now we're applying it to our HIV models.
Right.
We're also in our lab interested in a number of other disease models, cancer and other genetic diseases. So the fact that we can do it, as you said, is the key that we've done. Yeah.
And I think, as was brought out in the press conferences, that it is translatable to other areas. I mean, you're in the cancer research area, but it could be cancer. I know that's what Sharon was saying, so many other different applications, which it's exciting to see that.
Yeah.
So we're not only learning for HIV, we're learning for all other diseases.
Right.
This
Yeah.
can be applied broadly.
Right. And so I'm specifically in the HIV area, but the lab that I'm in is very broad. So my the principal investigator, Hans-Peter Kiem, is a hematologist and sees cancer patients on a frequent basis. But the work that we do is not just cancer. He has people in the lab that head up the cancer programs and people like me that head up the HIV programs. And we can use the same gene editing technology in all of those platforms.
So right now you're in animals.
Great.
And how soon is it before you get into humans? Is that something that's a prospect down the line, not too far?
We're almost doing it in parallel. So the work that we're doing is in a large animal model. There's work from other labs that's been doing similar strategies in mice. And the data in mice and the early data from us has allowed our biotech partner, Sangamo Biosciences, to get approval for a phase 1 trial of the same technology in HIV-infected patients.
So how do you see this moving? What are the next steps?
Well, right now what we can show is that the gene editing works, but the level to which it works is beneficial for some animals, and for some of the other animals it doesn't hurt nor harm them. So what we need to do is edit more, so edit a greater proportion of the cells, and also start to think about ways that we can select for those cells. So basically, in our case, we're editing a gene that HIV needs to get into cells, and we can get that into about 5% of the cells, and if we can boost that number up, then we could have more of an effect for the infected cells that come out and basically fight against that infection.
So it's just a matter of quantity at this point. Your
Correct.
quality is right. Do you feel that, and this is sustainable?
Yes.
I think that was one of the questions in the press conference. How sustainable is the product, or is the gene editing?
We're very lucky in that we're targeting a gene that is known to be mutated in a significant portion of populations in Northern Europeans and the Middle East, and populations that have gone around the world. So there's a lot known about people who are already mutated naturally for this gene. And there are some correlates that maybe there are a few susceptibilities are a little bit higher, but for the most part they're completely healthy.
And how do you introduce this into the cell, into the human,
so,
or into the subject, I should say?
Right, so the whole process is there's a gene that can be then made into RNA, and then that's made into the protein, and the protein is what's actually doing the job. So we actually take the middle step, the RNA, and we can introduce the RNA into cells, And what that allows it is that the protein can be made for a very short period, enough to target the gene and mutate it, and then everything disappears. So one of the big advantages to this strategy is that we edit the gene and then all the machinery is gone. So except for that edited gene, the cell should be completely normal.
Are there this would be considered an immunologic approach, or gene editing? It translates to an immune
Translates
approach.
to an immunological approach, yeah.
But is there other immune approaches that you would partner this with
Absolutely.
to make it more effective?
Right. So we're very interested in that. Like I said, at this point, the percentage that we are at, it's not going to be able to do the job on its own. And maybe one day it will, but for where we are right now, we know that we can boost the immune system. But if we can combine this with a strategy like a vaccination approach, one of the reasons that people think that vaccinations don't work so well for HIV is that HIV targets and kills the cells that are vaccinated. So if we can protect those cells with our gene editing strategy, then vaccines might work better.
Have you found anybody at the conference that was helpful to maybe seeing another poster or something?
Absolutely. There's a number of vaccine and antibody-mediated approaches here that we would love to work into our model.
Which could maybe be the real big reason you showed up here.
Yes, yeah. Making connections and thinking about where people stand as far as wanting to combine their approaches with ours it works really well because there's a lot of other non-human primate researchers here, and they've already sort of validated their model, we validated ours, and bringing them together is pretty straightforward.
It's exciting.
Yeah.
It's exciting. I thank you very much. I thank you very much for being here,
Yes.
and also for showing up to do this interview.
Thank you very much.